Welcome to the 6th Annual Soilcraft Conference. Join us as we listen to world-class speakers in the space of regenerative agriculture, as well as human health. We grow food. Food should nourish us, and often we find that our modern food is laden with unwanted synthetics or void of the minerals and vitamins we need to thrive. We endeavor to host this conference for your inspiration and education. Our calling is to help you produce food that heals people and our planet. Join us in this journey to become better stewards of our bodies and the world we abide in.
All right. So, you could rinse this off, but you don't need to. You can blow it off a little bit. That'll make it a little bit easier. But you actually don't want to remove a lot of it. You want to leave enough there to see the characteristic of its interaction with the organic matter and the soil around it. I'm not even going to cut it off. I'm going to make sure it gets flat enough, and then I'm going to get it in there. Well, maybe I'll cut it off. We'll see. It's chunky. But this is like how sophisticated we're going to go. We're not going to wash it. We're not going to stain it. We're not going to do nothing. We're switched over. Epic. All right, so I'm going to do a couple different things. Number one, these things are awful. Thank you. Interesting. This might be misaligned. Just give me a second. There we go.
Chem-Fallow, or if you had a Brassica that's not friendly to that, where does that put you?
Say it again. We're there.
Multitask? We live in a dry area,
Mm-hmm.
so we'll Chem-Fallow. So with a
Chem-Fallow, that's the thing I don't understand. What's Chem-Fallow?
Just keep anything. Chem-Fallow is just to keep anything from growing there to save moisture. That's our chemical.
But what does that actually mean? What are you doing to get that?
Glyphosate.
Woo!
I know. We're moving, putting humics in to try to calm it down. But
Yeah.
moisture is so important. We seem to think or compare it to say you have a Brassica that's non-mycorrhizal vascular friendly. So what happens to that?
So if you just had all right, so there's our mycorrhizal fungi. If you guys get the mixes from people, almost always the ones that are commercially available have trichoderma in them because they know this. They knew this 10 years ago, and they just put it into the mix. So you just need trichoderma to be in your mix. People buy pure trichoderma all the time now for plants. So that's what I would do, so your Brassica crop will be great.
Okay.
So
What about the year when nothing's growing there?
what's the logic behind that?
Try to have some moisture so you can raise the next crop.
It's a farming practice for conserving rainfall, giving the soil a rest for 1 year. So in 2 years, you have one crop, and then it becomes fallow. Well, in the olden days, still, you used to till it and fallow it by using rod weeders and just grabbing weeds. But now people are starting to spray, and they call that a chemical fallow or a Chem-Fallow.
Yeah, so that's an oxidative stress pathway, right? And so you'd be drying it out and making it more alkaline anyway. So I don't see that as a pathway to doing that. What I would do is I would do a crop that you terminate early, like a cover crop that you terminate early and crimp, and let that ride into the next year, and you'll have a better result. The idea that every other year you're adding that much glyphosate, how much glyphosate is in your product? Because you know that Moms Across America is testing that and then showing that it's affecting kids and it's in school foods, and people are very savvy to this, and so there's going to be a market demand reaction. So just economically, I would try to transition and find something else. Yeah.
Is that still there?
What do you mean? Later? Oh yeah, depending. All right, so in Texas, in Houston, where it's high humidity and it's hot, these things break down much faster. in the colder, more temperate climates, a lot of these things can last. And so we need to bring in compost, like microbes, to break these things down. But we also need to test. And luckily, there's now ability to test product for glyphosate. But just test. Because, I mean, if you've taken care of it, you've taken care of it. I mean, that's the whole point with all this stuff, is if we don't test, we don't actually see. So, and the direction we're headed is MAHA. We're going to be testing. We're going to have citizen laboratories everywhere. I'm going to be helpful in setting them up. You know? And, I mean, it's going to happen. So, we're here with this, with a root. It should be like that. Something. Okay, that's real. Now it's not. It'll come back. And now we're real. Okay, so there's a weird oscillation with the colors on the projectors. So don't use a projector. Use a 4K screen because it's a 4K camera. It'll be a lot cleaner and clearer. It has that correct color. It has the evidence of it going around the roots. We could travel more into it. Do you guys have a screen, like a screen or a TV we could connect to it for tomorrow? That might be better.
We'll figure it out.
Okay. Do you guys have questions?
Any questions? Hey, well, that was fortuitous.
Just for clarification on that gentleman's question on the mycorrhizal fungi, the spores living in the soil, how long can they live in the soil for? So say you inoculated a field, it grew, and then you harvested that crop. How long can those spores stay in the soil? Like you seed your next crop into it, are they there and active for the next crop?
The spores don't immediately germinate. So spores are insurance policy. The root fragments, the propagules, that's the reason why Mike Amaranthus, mycorrhizae.com, the godfather of all mycorrhizal fungi inoculants, he was the scientist who created it and then created the company. It's the root sections, the dried root sections from the prior year that are the thing that makes the difference because they readily partner with roots. We don't know when the spores actually germinate. And spores can last a really long time in the right conditions. But in the wrong conditions, they're cycled right out. And so that's why I also say we should test your soil and your roots with something like this so that you can see this. And we'll do more. I'll keep working on it. There's other plants there. The squash plant is probably going to be better. Squash roots are typically larger. And so you get a better picture. How much time do we have?
Lots.
Oh, good.
So
I'll
can
do that.
you can
I'll do
riff.
squash
Let's
next.
riff. Anybody else have any questions?
Yeah.
Come over here.
And we got to get you.
Thanks, Matt. Very informative. And so one thing you mentioned is that mycorrhizal is not present in compost. Mm-hmm. And that's simply because of the absence of roots. Is that
right?
correct?
And so that's why there's been a new thing where people are planting plants that are inoculated into their compost to finish it so that they can say that there's mycorrhizal fungi in it.
Gotcha. Now, can the spores be present in the compost? And once you inoculate your seed, your cover crop, whatever, you go and plant that, and you develop a root, can the mycorrhizal then come?
Not from what I've seen. Because those things are the food source for your soil food web. And then for your soil food web to actually do its work to make the inoculant or the inoculum of compost to be ideal, it's 6 months to a year. So you've cycled through those things. It's the food. I think this...
Over here?
Okay.
Oh, I missed you last time, so I'll get him first.
Two parts here. So, so you are on the search for bridging organisms, so like Trichoderma,
Yeah.
those kind of things across the board, and potentially we'll find that the beets and chard will have some
I
sort
hope.
of arrangement with that. So, what types of soils are you pursuing as far as in matrices? Are you going, first horizon? Are we going deeper with regard to trying to, wake up those types of potentially dormant types of endophytes that might persist in those clines?
Oh, it's interesting. I don't think that they're deeper. Microbes tend to be in the top 6 to 8 inches. Same thing with all the nutrients. And you can, but at the same time, the deepest holes that have ever been drilled into the earth have microbes down there. Even though they have like crystalline diamondized water and all this crazy stuff, there's still microbes. So,
Great. And then the second question is, with regard to endophytes and the transfer of information and data, you talked about gene transfer and those kind of things. Is it going broader than what you're communicating with regard to insects, and maybe now we're talking about mammalia, to transfer genes from plant to plant based on contact with an endophyte on one plant, transferring it to another?
So that's really, really awesome. So none of us are isolated. We are in constant communication with the world around us, and organic matter having DNA in it actually gets eaten by bacteria, and they use that DNA. And they've also recently proven that plants get DNA from bacteria, and they use it too. And so it's not that they're using it and consuming it. No. They're adopting it. They're absorbing it and using it, which means also that the same microbes that are in our gut are absorbing information. And we all know this. I mean, remember the talks of Bruce Lipton and all these people, epigenetics? They're like, well, there's actually very small molecules in your food that affect your epigenetics, right? It's all part of that. And so our gut microbiome absorbs DNA from the microbes that are in the food that we eat. And if you believe that those microbes in you, and they are you, you're not like filled with foreign matter, it is you. Like we are the bacteria. It's not that whole thing where like, you're not even human. There's all this bacteria inside you. Like, that's you. That's not like not you. So it's like really important to recognize that we are constantly exchanging information with the world around us, with what we breathe, what we eat, and what we absorb through our skin, the light. Everything is a feedback loop with our bodies, our senses, and the microbiology within us.
How specific are mycorrhizal to individual plant species?
Well, like I was talking about, how Enterorrhoeaceae is dominant, it partners with all those 90%. That 90+%, some people say 95%. But they partner with all of them. It's just that there are specialized ones that are more for different pHs, more for different soil types, and conditions, like high salinity, or sandy soils, or calcareous soils. There's specific mycorrhizal fungi for those, and that's actually in the book out there. You guys can all feel free to like look at the books and see what's in them. You can open them and peruse, because there's a lot of information that once you're like, oh, that's the actual fungi I want, and then you just Google it and you can buy a culture of it, and then you're like, I'm going to scale it myself. And then you have it, and then it's permanently in your soil, and you just did it as a project one year, and now it's done. It's just that simple.
Just looking at the root under the microscope,
Let's get in.
and the value of that. So when we see that activity on the root, you see the glowing.
Mm-hmm.
Is that telling us that there is mycorrhizae in the root and that's active?
It's not very active, or there's not like phosphorus in that soil medium. I don't know, but it's not bright like the examples I already showed you. So like if we were in my classroom, I would be like, this looks like it was doing well and then like ran out of phosphorus, or is just beginning, because you're seeing those edging around the cells, but there's no like the whole thing glowing. That color when it was doing its wave, it doesn't seem to be doing it now, which is bizarre. But that wave thing that it was doing when it was really bright, matching what I've already had, that's more definitive, because then you have the buildup of phosphates. The reality is it's like you could have something, and if it's at a low level, then it's not efficacious. So you might as well, it's not even like there at an efficacious level. So it's, people all the time say like all soils have everything. At what rate? You know what I mean? And so like I wouldn't say that this is a definitive like inoculation yet, because I want to see the root 80 to 100% glowing solid like we saw, and then developed successional levels and representations of the life cycle. So it's been here a while, it's doing this work. I can tell that this is that fungi. I can see the evidence of its effect. And that because the way I was trained by mycologists, I really lean into the non-definitive. And also it just benefits us. It's like, you don't want a mediocre thing and be like, yeah, it was great, it was mediocre. No, you want it to look, because it's supposed to be 80 to 100%. So it's like, it should just be there. And if it's like sort of there, it's like, we got to do that inoculant again or get a different inoculant.
So as a grower, what is that telling me, though? If
You might need to re-inoculate.
I need to inoculate, or that it's there
And it's beginning.
and I don't need to add any more.
Right. So from here, I would wait. I would wait like 2 weeks, and I would also do a soil test on the soil, try to understand what's going on there. But there's other plants, and I would do more than one test. That's really important too. Plants are individuals, and we've got winners and we've got losers, and the winners we save seed from, right, in our gardens. And so I always encourage doing more testing. I can play around more, and in fact, we can just switch things up. So what is this growing medium, Zach?
Isn't it amazing that we have tools like the Microscope Matt Powers recommends? There are so many excellent tests out there to bolster your microscope work or add data points for a full analysis. If you want to learn more about what tests we trust, reach out to us. We utilize several soil tests as well as microbial bioassays and plant sap analysis. At Soilcraft, we like to work on as many quantitative and qualitative data points as possible so we can make a precise and confident decision with you. Call, connect, follow.
Then one more question that I would have is, if compost does not have mycorrhizae in it because there's no living plants, then what about soil that doesn't have a green bridge from one crop to another?
It definitely starts oxidizing out, and you start losing things, but unless you're testing and see that it's gone, it's hard to say. It's doing it again. Do you see that? All right. We're just going to move quick. So I'm seeing potentially hyphae right there. And this is like a phosphorus crystal. That could be evidence there. So the resolution, I always have a 4K screen right here. And yeah, that looks right. Yeah. All right, good. Here we go. You can see it here. I have to ride in and out of it, but you can see it there, and then going down, and it's initiating along there. And the way it's blowing up like that into it, that's the arbuscule. Those right there. So it's just beginning. You can see a thread of it running through it, all the way through it. And as it heats, it's curling the root, so that's part of the why it's also being annoying. We go to where there's a little bit more interesting things happening. Oh, I'm off. There we go. Yeah, it's important to have all the pieces of equipment. Usually I don't use my cell phone to block the bottom light. I use a piece of felt. And this tray is really weird. But yeah, I would want to see more definitive science. Wait. There we go. There we go. But yeah, see how it's establishing there? Just like we learned. You guys see it now? It's establishing. It's just beginning. You can see it there. You see the pinpoints of it, of delivery there too. You see that? Right there. So yeah, it's just beginning. But thank you, Zach, for doing that. I'm going to give a round of applause for Zach for bringing that in. Thanks, Zach. So what he did was he put inoculant on seed and did a germination test. We all know seed germination tests take 10 seeds, put it in a damp paper towel, put it on top of the fridge where it's a little bit warm, right? Added a little bit of inoculant, and then you can check that inoculant's efficacy on the seeds that you just are about to plant, right? But you can also look at the actual inoculant underneath it, as we can right now. And there's two ways that you can do that. You can physically look at it, but that's going to be pretty blinding. Or you can break it up a little bit and put it in water. Am I on time now? Anybody have, yeah. You got me here, guys. Ask the questions. The thing also is that everyone wants like a number, and it's easy to know 50 to 100% or almost 100% when we're doing this, because our eyes just are designed for that. We're really designed to see amounts. Like if you're going to split something with your friend 50-50, you're going to know your half, right? And you have brothers. If you're going to split something 50-50, it's going to be 50-50, right? And so, like, we're good at this. And so, just being able to eyeball, mycorrhizal fungi has minimum 50% inoculation for it to be, like, actually taking. So it's a full inoculation is 80 to 100%. So it's very easy to verify all this.
Hey, Matt, we talking about mycorrhizal. That's great. But what really caught my eye is the nitrogen. I'm a wheat farmer, and we spend a lot of money on nitrogen. And I don't like spending money on nitrogen. But you were talking about TeraGanix. Could you share a little bit more about that?
Oh, yeah. It's EM. Have you heard of EM yet?
Effective microbes?
Yep. So what that is yeast, beer yeast, not lager yeast. Lager yeast is for ecto. You're going to go ecto with lager yeast. But beer yeast, Saccharomyces cerevisiae, bread yeast, that is going to be the EM microbe, and it's an endophyte. It's in like all plants tested. And then it's lactic acid bacteria, lab. I mean, that's, rice wash water with the milk and all that, the Korean natural farming way of doing it. Or we're talking about water kefir, right? I mean, it's not that complicated. Water kefir, you guys, everyone in this room can take the grains and combine it with sugar and scale it up. You can do that to the 1,000 gallons too. So water kefir is lactic acid bacteria and yeast. That's what it is, right? And so that's like a primary part of EM. And then there's this other part that's provenance over bacteria. That's Rhodocytomonas palustris. That's a primary endophyte all over the world. It feeds 4 ways, but different ways within those ways. So it can photosynthesize and do all these different things. And it's fundamental to our guts, fundamental to so many life processes. You'll find Rhodocytomonas palustris inside plants. You'll find it actually in the chloroplast of the leaf. Like, it is wild. And so you're like, wait, they photosynthesize and they're in the leaves? What's happening? Are they also photosynthesizing alongside the plant? Yes. Unbelievable, right? So, which opens a whole other bag of worms, right? And then there's streptomyces in there. There's a bunch of other things that are in EM1. But those three are the primary ones, and they're the efficacious ones. They're the ones with the most written about them. You can literally look up R. palustris as an endophyte or biofertilizer, and you'll find tons. Lactic acid bacteria, same thing, tons. Yeast as well. Just tons of information. Saccharomyces cerevisiae is what you want to look up, though. It's got to be specific or you don't find that. So what does that do? Why is that good? Well, they're endophytes, first of all, but what are they specifically doing? You guys know the answer already. When a plant has excess sugars, what happens? Right? When it has excess simple sugars, what happens? The bugs come, right? We were reviewing that this morning. What does yeast love? Sugar. And up to like 45% of the CO2 for photosynthesis can come from internal endophytes, yeasts. So these plants are doing all this with microbes. The microbes are their digestion. So it's like, okay, all right, so the yeast eats the excess sugar, turns it back into CO2, starts the cycle over so that it can handle the excess. And then lactic acid bacteria, do you guys know what that does? So it's the thing that like you would take and spray on your chicken yard and remove the smell because it turns the nitrates into amino acids. Yeast can also do this to a degree, but it's really lab that does it well. So I have a student right now who has a feedlot, and they're using EM, and they just had a bunch of downer cows that they had to deal with. And usually in that area it smells forever. And they applied EM, and it didn't smell the next day. And it's just been good. And so now they're adding it into the water. They're adding it into the floor. And this is not hard. You could get, EM from TeraGanix and start brewing it up and extending it up. And the danger is, as you're extending, the purple non-silver bacteria, which likes pH 9 to breed, at pH 3.5, senesces out. But you can also just go to any pond store and order purple non-silver bacteria, Rhodocytomonas palustris specifically. Because the thing that eats algae in the overgrown ponds and our sloughs and our ditches and our runoff ponds that we have on our farms, all that algae, purple non-silver bacteria eats it. So you want to clarify that pond? That's how you do it. And again, you can buy these things at aquarium scale, like commercial aquarium scale, and then fix the entire pond, or mix it in with your water kefir brew, and then apply it and see a massive reaction. So the reason they can reduce the nitrogen is the plant themselves, if it's an endophyte, it must release nitrogen. And because they're transforming it from nitrates, because they're adding it to the actual fertilizer and then adding it in, any system that's oxidized is going to turn any form of nitrogen into nitrate within hours. So they're mixing it in, and they're literally turning the nitrogen source into amino acids, so it arrives as an amino acid, and the plant knows what to do with that. The plant doesn't have to expend energy, and so you're saving the plant energy, and because amino acids act as energy in the plant, they get a gain. So it's something that's so simple, but it's just bringing back the endophytes that are in the plant. And it's about rate, right? It's like it could be there, but is that at efficacious levels? And that's the real thing. It's not been mapped out yet, like the full amount of how much microbes you need or how much microbes you need for this crop or for that crop or for this soil, for that. And that's why right now you have people who are applying and they must test. Like if Ryan's going to do an application, he's going to test before and after. Why? Because he doesn't want to overdo it. He wants to do it enough so that it has the effect that he wants. And so that's why we're testing so much. It's why it's so critical we test, because you may apply the same amount as he does, but he gets there, and you're not there yet. And you're like, what? And you just have to add more, and suddenly you're there, and then the flywheel of momentum begins. So I soak my seed in EM so that they embody it, but I also do foliar spray. It absorbs really, really readily, and it has a seriously amazing effect on plants. But it's just because we're returning the plant like it's like gut biome. We're giving it like probiotics and prebiotics, right? And so we're seeing these incredible reactions simply because we're returning things to normal. We live in a miraculous world, and the mystery and the miracle of this world has been greatly diminished because nature has been greatly diminished. If we bring back like the actual power plant of the earth, photosynthesis, back online, we will see just such incredible reactions happen. And we'll see it on the small scale in our gardens. We'll see it on the large scale on our farms. And when we do this at like a systematic level, like if we actually have California, instead of burning their forests down on purpose, because that's what they're doing, I mean, think about it, guys. If you take all the water from the foothills, which are already pyrophytic, meaning they naturally have fires, you remove the water for 50 years, and then you run it through tunnels, and then do through turbines, and then steal the people's water as you charge them electricity on the water. A, you make everyone feel horrible, and B, you guarantee that there's going to be massive fires. Massive fires. And anyone with half a brain can see it just playing out. You go out to the creeks in California, the foothills, and it's 20 feet deep cut into the decomposed granite. And water never, ever reaches more than like a trickle. And you're like, where's the water that cut this? Where's the water that like fed all these trees and all, and it's just gone because they took it from above. Do we have time for one more? I can keep going on California. Woo!
I was just hoping you
Sorry,
could kind of
Californians.
elaborate on like the illumination of the mycorrhizal fungi and maybe clarify like the physical process that's happening with this microscope. Like the blue light, is that illuminating just phosphorus that we're using as like a proxy for the mycorrhizal?
So if you go and buy like a cheaper version of this, the bell curve on it's wide. So it'll hit other things and other things will glow.
The bell curve of like the frequency?
Yeah.
Okay.
So this is a pinpoint. So this is 489 precisely. And I know this because I had a light that was like 487 to like 487 to 492, and it was a mess. And I'm like, why can't I get this to work? And the guy was like, you need to send me your thing, and I'll test it. And I'm like, I can't send you this through the mail. It's $3,000. And so I never sent it to him, but then I talked more with Mike Thomas at LW Scientific, and it's a very specific like spike. It is not a bell curve. And because of that, we can see very precisely the autofluorescence of mycorrhizal fungi. And the reason it's autofluorescing is you can't find this in the mycology world, probably because they're too scared. But in the medical world, for over 40 years, NADPH is a molecular biomarker. It autofluoresces. It's established. And the thing that mycorrhizal fungi, if you actually get into the literature, mycorrhizal fungi and NADPH, you can find it. And that's the main thing that's glowing. And so it is a phosphate that is glowing. The light that it's hitting at is causing the molecules to excite. And it's this emission that we're seeing that is the light. And so if we put different things under here, like if we put chlorella under here, it would be blood red. Because all chlorophyll comes red through this. And then there's a filter cube. So as it passes back up, it cuts it off at 510, so you only get certain bandwidth of light above that. So it's very, very, very clean in terms of image because of that filter cube cutting off at 510. So we're just seeing the emission. So it's a really clean image. It allows us to identify things. And there's, as you saw, lighter green. That's because that's lignin. And lignin does autofluoresce, but when you hold phosphorus up to lignin, and you know it's lignin, the colors are totally different. And we saw it. One's lurid, like almost yellowish highlighter green, and then the other's lime and see-through. It's like the 80s lime green. Do you remember how everything was black in that lime? It was great.
Good man. Matt, thank you very much, everybody. Matt Powers.
Thank you, guys.
We're so glad you enjoyed this installment from our in-person conference. We want this to reach as many people as possible, so we've decided to release this free to you bit by bit every month. But imagine how awesome it would be in person. The networking, questions, and discussions that result are equally as valuable. Join us next year in person for our 2026 conference. Your presence makes the experience. If you have any questions or needs, please reach out. Call, email, follow. We can help.